Meristematic tissues retain the ability for cell division for a long period, participate in the formation of all permanent plant tissues, and determine the plant's sustained growth. Cells of meristematic tissue are thin-walled, polyhedral, tightly packed with dense cytoplasm, a large nucleus, and very small vacuoles. They are capable of dividing in various directions. Based on their location within the plant, two main types of meristems are distinguished: apical and lateral. Apical meristems ensure the growth of stems and roots in length. This growth is called primary growth, and the meristems themselves are referred to as primary meristems. The primary meristem constitutes the seed embryo, and in an adult plant, it is preserved at the tips of roots and the apices of shoots. In addition to apical meristems, primary meristems include those lateral meristems that were formed from the apical ones, but have not lost the ability for further division. These include in particular photoderm, which forms the dermal protective tissue, ground meristem, which gives rise to the system of ground tissues, procambium, from which primary vascular tissues or bundles are formed. In monocotyledonous plants, the procambium forms the tissue of closed vascular bundles. In dicotyledons, a narrow strip of procambium cells retains the ability to divide and forms the cambium, a secondary lateral meristem that ensures the growth of stems in thickness. Cambium cells can be seen on tree cuts or stumps in the form of cambial rings. Besides the cambium, another lateral meristem is the phylogen or cork cambium. It forms the plant's secondary dermal tissue, cork. In addition to apical and lateral meristems, two other types of meristems are found in some plants. Intercalary meristems are preserved as separate sections in zones of active growth, for example, at the base of internodes and leaf peritals in grasses. Wound meristems form the sites of tissue and organ damage and give rise to callus, a specialized parenchymal tissue covering the injured area. Disorganized growing callus tissue consists of cells that divide chaotically and indefinitely. Theoretically, each of these cells, under appropriate conditions, can give rise to an entire organism. The tissue culture method is based on this property.